Okay, no.
Well, in the interest of giving Nick as much time as possible to tell you about his dissertation research, I suggest we get started. So, it's a really big day. Really happy to be here. Very happy to see all of Nick's great network of supporters and committee and colleagues here. It's fantastic. And I just want to keep this very short, but I want to say that, just Nick has really, he's worked very, very hard. He's got an incredible work ethic and has done really an amazing job of really expanding our, basically the scale and scope of knowledge of coastal biodiversity in the Northeast Pacific with his dissertation research. And he's really moved the needle on our understanding of the processes, species, and factors that make up that biodiversity, that affect that biodiversity. So I can't start this without a little slideshow of Nick, because it's been quite a great journey. But anyway, Nick has been really integral throughout, from the beginning of this, in developing questions and designing studies to address them in this fairly dynamic and relatively challenging coastal ecosystem of sandy beaches. One big aspect of this is it's not something you do by yourself, so he's an incredible team leader and team builder, and he has mentored so many undergraduates. I meant to try and get a count, but it's probably over 100 in his period here. The quantitative sampling of beaches, he says here that I sampled 60 beaches, over this great latitudinal scale, but what he doesn't say is that it really requires moving a lot of sand, core by core. So if we think about Nick's dissertation research and how much sand he had to move for it, he had to move, so each beach is like a metric ton of sand. We fill up these bags, they're about 50 pounds each, 45 bags per beach, metric ton of sand. So that's about 12 dump truck loads. So picture that in this room. And he sieved it all through very fine mesh. So this is an incredible accomplishment. But, and sometimes this was in challenging conditions. So here he's taking cores in the active swash zone. And here they're rinsing. In the active swash zone, you can see surfers getting wiped out in the background. So, yeah. And then, so the goal is to get all this material, get the sand out, get all the material literally into the bag. And these gallon Ziplocs, we could also add up how many gallon Ziplocs we use, but we're not going to. But that is just the beginning of the work. There's a painstaking process of sorting, identification, and then data analysis. So this is not a lightweight undertaking. All right, but throughout this journey, Nick has pulled far more than his weight. He's been an incredible colleague and graduate student, and I want to give him a lot of time to talk, so I'm not going to go on and on, but I just have to say that he's now sort of expanding the boundaries of our knowledge yet again into the surf zone. So I will leave it with that, and I'll turn this over to Nick.
Thanks for that introduction. Thank you all for coming today. I'm finally excited to share with you some of the pieces of my dissertation that I've been working on. In the short time I've spent working on this, I've explored spatial and temporal patterns in the biodiversity of coastal ecosystems using sandy beaches as a model ecosystem. So biodiversity is pretty broad. It's kind of every little thing, living thing you see in this picture. It's the variety and abundance of species in a defined unit of study. It can be measured as species richness or the number of species, but there can also be a component of abundance, so how abundance is spread across the species present. I'm interested in the structuring of ecological communities, so the patterns in biodiversity and the processes driving those patterns at the species level. So species are added to a system via speciation and dispersal. And the relative abundance of these communities are shaped by abiotic and biotic processes and ongoing dispersal of organisms. These processes can act on biodiversity across a range of spatial and temporal scales. So for example, disturbance and including human impacts can affect these processes at the global, regional, and local scale over centuries, decades, years, months, and weeks. So evaluating the responses requires comparisons across relevant spatial and temporal scales, which can be challenging given the availability of data sets, time, and money. My research is also motivated by concerns about biodiversity declining and the alteration of ecological communities, and a need to catalog and monitor species and ecosystems in the face of climate change and human impacts. Nowhere is that more apparent than at the coast. With 41% of the world's population living at or near the coast, growing human populations have been considerably impacting these coastal ecosystems. Open coasts are dominated by sandy beaches compared with subtidal and intertidal rocky ecosystems that are studied extensively. Beaches are considered to have relatively low biodiversity. However, beaches support unique biodiversity that's important to ecological functions such as nutrient cycling, wildlife support, and coastal protection. Sandy beaches are highly dynamic soft sediment ecosystem exposed to the waves and currents, which makes it challenging for plants and macroalgae to attach in these habitats. Beach communities are strongly influenced by the physical characteristics of a beach, such as the sediment and wave climate, which can vary spatially and temporally. With little to no in situ primary production, two main trophic subsidies, macrophyte rack, pictured here, and surf zone phytoplankton support beach food webs. These two subsidies can vary in space and time. Subsidies provided by kelp forests and highly productive coastal regions make California some of the most biologically rich beaches in the world. However, we know the number of species, abundance, and biomass can vary in space and time. And just to give you an idea of what that is for beaches, on average, we have about 27 species per beach survey here in California, and that can get up to about 50 or so, especially at the beaches around here. On average, about 60,000 individuals per meter of shoreline. That can get upwards of 300,000 and about 10 kilograms per meter of shoreline as well, and that can reach up to 80 kilograms on some of the really productive beaches. So they're an ideal ecosystem to study these processes. You can partition beach macroinvertebrates into two groups based on functional traits, trophic gill, dispersal ability, and intertidal zonation. We hypothesize that these differences could influence their biodiversity responses to varying spatial and temporal scales. So these are two groups that come up a lot in this talk, so I'll define them here. On the right are examples of lower beach species that inhabit the mid to low intertidal, and they're based around a phytoplankton food web. And those include species like polychaetes and crustaceans and some mollusks here. These species can disperse long distances, possibly facilitating recolonization following a disturbance. On the left, we have the rack-associated species. They inhabit around the high tide line and upper beach. They are based around a kelp food web. And these include species like insects, the beetles and flies, and crustaceans like the beach hopper and isopods pictured here. They're an important component of the beach. They make up about 40% of biodiversity in California and are characterized by low dispersal and direct development, which makes them vulnerable to human impacts and climatic impacts, which I'll hopefully show in this talk. My dissertation focused on patterns in biodiversity of sandy beaches on a variety of scales across the state of California and responses to that biodiversity to human impacts. First, I'll start with the results on the biogeographic patterns, then move into temporal trends, and finish it up with impacts associated with urbanization. Biogeographic units such as the 4 bioregions pictured here in the colored boxes and the provinces divided by the dashed line there at point conception are geographically distinct ecological communities that have similar sets of species and environmental conditions. So boundaries are often associated with physical and oceanographic barriers between these regions. And research has shown that sea surface temperature is the most commonly referenced factor influencing the biogeographic structure of marine communities. This includes rocky intertidal ecosystems that have been shown to conform to these patterns along the Northeast Pacific. For this chapter, I was interested in describing the biogeographic patterns and drivers for sandy beach macroinvertebrates in California, comparing them to existing biogeographic patterns for marine communities and exploring how they differ for those 2 different invertebrate groups that I described earlier. To answer these questions, we surveyed 35 beaches from San Diego to the Oregon border from 2009 to 2015. These are indicated by the yellow circles in the map here, and they span all of these provinces and bioregions. Because it's beaches, we have to talk about littoral cells. They're distinct compartments of the coast that can contain a complete sediment cycle. So the sand comes into the beach from the rivers or bluff erosion, it's moved down the shore via waves and currents, and then it finally exits the system through submarine canyons or blows into a dune field. These are important to beaches because they influence the quantity and quality of sediment and habitat, and they could potentially influence ecological communities, but that's not yet been explored, and what we'll do here. California coast has been broken into 25 littoral cells, indicated by the dashed lines and numbers here, and our sites are distributed across 13 of those littoral cells. One way to describe biogeographic patterns is to use a multivariate approach to look at how similar or different communities are. So we use non-metric multidimensional scaling and Permanova to examine community structure for beaches in different biogeographic units and littoral cells. If you're unfamiliar with this approach, basically each point on the plot here on the left represents the beach. The closer the points are together, the more similar the communities are. And you can use Permanova to determine whether clusters of beaches differ significantly. So to orient you to the plot and what we're looking at, littoral cells, I know it's a little confusing, there's a lot going on. Littoral cells are indicated by the different symbols, bioregions by colors, and provinces by open and closed symbols. So as I said, there's a lot going on in this plot, but what I want you to take away is that the study beaches belonging to the same biogeographic unit or littoral cell clustered closely, and those clusters differed significantly. That means they conform to existing patterns for other marine communities in the region. And also, littoral cells appear to be an important biogeographic feature. But if you look at the lower beach and rack-associated species, the 2 bottom plots, they don't really differ that much, maybe a little bit more spread out in this analysis, but we didn't really see a big difference between the 2 groups. However, using this analysis, they did differ. So this is the distance decay of similarity relationship for lower beach and rack-associated species that basically shows the degrees of isolation across space. So similarity values that we took from the last slide, which were how close or far apart those dots are plotted as a function against the distance between the study beaches. So sites that are closer should be more similar, and you should have a negative relationship here. The steeper slope for the rack-associated species suggests higher degrees of isolation for the rack-associated species than the lower beach species, possibly because of their limited dispersal ability. So what are the environmental factors influencing these patterns? We chose factors that are generally important in structuring both marine communities and sandy beach communities. Sea surface temperature, salinity, and productivity are the strongest predictor of marine biogeographic patterns. The resource availability and habitat characteristics that we chose are more specific to beaches, such as sediment and wrack, and have been shown to influence community structure. We use a distance decay of similarity model to identify environmental factors that significantly predicted variance in community structure among the study beaches. What these results show is that we explained a high proportion of variance, so 43 to 59% of variance in community structure. Oceanographic variables that are in blue in these pie charts are also are the most important, but general characteristics, so the habitat characteristics and resources, are almost as high a percentage as sea surface temperature and salinity. So the importance in the beach-specific characteristics differed between the lower beach and wrack-associated species. For the lower beach species, the most important ones were habitat characteristics such as grain size and beach width, and then for the wrack-associated species, it was drift macrophytes, so wrack. Our results also revealed the importance of donor ecosystems and wrack subsidies in structuring sandy beach macroinvertebrate communities. So this is supported by the figures on the left showing from top to bottom that the species richness, abundance, and biomass of wrack-associated species was strongly correlated with drift macrophytes. A compositional shift in the dominant kelp species at Monterey Bay may also represent an important factor structuring macroinvertebrate communities on sandy beaches. So Nereocystis, an annual kelp pictured up here, is dominant north of Monterey Bay, but you can also find it down south in some of these beaches, or in some of these kelp forests. And then Macrocystis is dominant south of Monterey Bay, but it's found all up and down the west coast in North America. The different colored symbols in these plots represent where we found those species on the beach. So yellow is where we found Nereocystis, brown is where we found Macrocystis, black is where we found both, and white is where we didn't find any. And the shift in dominance might explain the differences that we see in this MDS plot that I showed you earlier, and I switched the colors around a little bit, and explain it with that sort of spatial variation in the composition of dominant kelp species north and south of Monterey Bay. So highlight from these chapters are that the biogeographic patterns for beaches conform to existing patterns for other marine communities. Our results also suggest that littoral cells might be an important biogeographic feature because of their influence on habitat quality and quantity. Lower beach and wrack-associated species differed in the spatial structure, so wrack-associated species were more spatially structured than lower beach species, and oceanographic and beach-specific variables explained a high percentage of variance in biogeographic structure, but the main ones driving the wrack-associated and lower beach species differed between those two groups. The processes influence biodiversity occur at a range of temporal scales and often go unnoticed unless longer data sets are available. So the next chapter addresses questions regarding temporal change in biodiversity using an older data set from the 1970s. She liked that last chapter. So in looking at multi-decadal trends in biodiversity, I wanted to know if we can detect ecologically significant differences in the biodiversity across these temporal scales, and if so, can we identify the drivers contributing to that change or that are consistent with that change? So we've looked at climatic and anthropogenic ones like the ones listed here. To do this, we compiled an ecological and physical history of sandy beaches in Central and Southern California, and this woman here, Dale Strawn, is sort of the master of this data set. She started collecting this with a group in 1969 following the oil spill, and it continued for about a decade. They sampled over 60 sites and took over 450 surveys, so it's a really incredible source of data. However, there are challenges in compiling this data. So I got all these boxes from a garage in Whittier. I'm picking through them with undergrads, and I'm trying to piece pieces together. As you can see, the handwriting is illegible in some of these pictures, and species names are changing, and there are duplicates, and there are things missing. So it was a real challenge to work with this data. I had big plans for it, but what we came back doing was just using the cumulative species list from each site, because that's really what I was confident with in the data. And it's kind of a funny story, because I showed this slide at a conference that I went to in South Africa, and this woman here, Peggy O'Donnell, comes up to me afterwards and she goes, so that data sheet that you were showing with the bad handwriting, that was mine. And she's from Australia, and she worked in high school with Dale Strawn in Pasadena on this data set. Had no idea I was going to present on it. So, funny little story there. So while we were compiling all this data, we resampled a subset of 13 sites. We did 35 surveys in the 3 years that I was working on this. I like to show these two pictures because they give some examples of that kind of change you would see on the beach. So Scripps on the left had no seawall in the 70s, and by our 2009 survey, they had put in a seawall at some point in between there. So these are the study beaches ranging from Morro Bay to San Diego. Sites were surveyed 1 to 6 times during a 3-year period. After compiling all the 1970s data, we evaluated patterns and trends in species richness, community composition, and beach characteristics. I tried to recreate the photo here a little bit, but the stairway is a different direction. A lot of ecological change can be explained by the physical characteristics of the beach. So beach characteristics measured in the 70s were limited. So unfortunately, they didn't collect any data on rack back then. It would have been very useful. But we were able to look at change in intertidal width and grain size. So the sites from left to right are north to south to illustrate lack of regional patterns and change. We found significant differences at 3 sites, but what I really want to point out is the largest proportional loss in intertidal width was at Colwell Point right here, and I'll come back to that later. Because species richness is strongly influenced by sampling effort, I had to develop a calibration to adjust species richness for area sample, and I'll walk you through that here. So we used a program called Estimate S that has species richness estimators, and one is this extrapolation approach. So we extrapolated from a sample-based rarefaction curve to a larger area. So this is what we sampled to, and we were able to extrapolate all the way out to here with 95% confidence intervals that are unconditional, so they're open at the end here. From there, we plotted the cumulative species richness and area sampled from the 1970s, so what I was confident using with that large data set that we had that was all over the place. And we compared it to the estimated richness from this extrapolation graph here to evaluate differences in species richness between these 2 sampling periods. So for this example, it was 33%, and it lays outside of those 95% confidence intervals, so we deemed it was significant. So the results from all our beaches are presented here is the percent difference in species richness for the total community and rack-associated species. Although multi-decadal changes we detected in biodiversity for the total community and rack-associated species varied in magnitude and direction, with no regional patterns, as you can see from left to right, north to south. Our analysis revealed several results related to the history of human impacts and beach alteration. And those are indicated by the colors in this figure. And I'll go over those one by one, group by group, in the next few slides. Also, a large component of these differences were driven by rack-associated species in the bottom part of that graph. The majority of beaches with no detectable beach alterations during the study period, shown here in green, increased in species richness. These range from 20% to 80% increase for the total community and about 5 to 230% for the rack-associated species. The one exception was Coal Oil Point that I talked about earlier in the white bar here. Coal Oil Point, we observed a 38% decline and 62% decline in total and rack-associated species. Although there were no direct beach alterations on this beach, results suggest that there was a loss of habitat that would be consistent with erosion from sea level rise. Particularly important for this beach because it's bluff-back beach, so it has nowhere to go as sea level rises. During the time period, we had about 1 millimeter rise in sea level over that time span per year. And obviously the biodiversity response matches that because it dropped down, particularly the loss of upper beach and rack-associated species. This pattern differed for beaches that experienced continuous impacts commencing before the 1970s and continuing through the survey period. These are indicated by the gray bars here. Despite change, the species richness was consistently low at these beaches, so under 15 or so species in both survey periods. We observed 45% higher total species richness on average for these beaches for the total community, but for the lower beach community, or for the rack-associated species, 41% lower for 3 of those 4 beaches. And I'll explore these a little more in my next chapter, these types of impacts. ORV, off-road vehicle use, has been shown to cause significant impacts to sandy beach ecosystems, particularly upper beach and rack-associated species. Our results were consistent with recovery from off-road vehicle use at 2 beaches pictured here in blue. Recreational off-road vehicle use was banned from most parts of Oceano Dunes in 1984 and Arnold Road in 1992. Since then, we've seen an increase in the total and rack-associated community, and the return of low dispersal rack-associated species at these beaches suggests recovery is possible when you change management and outlaw major impacts like this. So in summary, the highlights from this chapter include that local scale processes exerted stronger influence than regional scale processes on biodiversity. And ecologically significant differences can be masked by comparisons of overall species richness. Although we found important declines in richness associated with sea level rise and erosion at one beach, we also found ecologically significant declines in rack-associated species at numerous beaches that represent a threat to biodiversity and function of beach ecosystems. We also found that changes in management, such as off-road vehicle use, can lead to recovery. And importantly, I learned not to make fun of someone's handwriting in a talk, because they might be in the audience. Human impact can vary spatially and temporally on the coast, so multi-decadal trends that I just talked about suggest that impacts associated with urbanization can have lasting and expanding impacts on sandy beach ecosystems. So this has led us to ask several questions about the impacts of mechanized maintenance regimes on sandy beach ecosystems. So far, I've demonstrated that California beaches are some of the most biologically rich in the world. That has a lot to do with them being subsidized ecosystems and huge kelp forest beds offshore that support these ecosystems in the very productive water. Also, we have these littoral cells that are influencing the biogeographic structure in the region, but these beaches are also sensitive to impacts. Coastal urbanization is widespread. You can see the light concentrated near the coast in this photograph, satellite photo from the US. And if we zoom in on our region, down in Los Angeles here, almost the entire coast is lit up. So this is a heavily urbanized and developed region, as I'm sure you all know. Beaches are highly valued for their socioeconomic importance. Over 150 million people visit beaches in California each year. Beach economies bring in billions of dollars and hundreds of thousands of jobs. Beach humans can have dramatic impacts on intertidal ecosystems. Urbanization fragments and degrades habitat, impacts biodiversity, homogenizes species composition, and alters ecosystem function. Coastal development has influenced the coast of Southern California for over 100 years, as is demonstrated in these two photos from Santa Monica, and urbanization is expected to continue. Development has led to the damming and channeling of rivers, which alters sand dynamics, and in response, we've nourished and filled beaches and armored the coast to protect shorefront property, which alters the literal transport that I was talking about of sand up and down the coast. Beaches are also frequently groomed to remove trash and macrophytes and wrack. In Southern California, 80% of the coast is beach, and 40% of those beaches are mechanically maintained, generally beach fills and grooming, some pictures here of those. For the purposes of this study, I define an urban beach as one influenced by anthropogenic processes, and they're subject to mechanized maintenance, so both beach filling and regular grooming activities, sometimes multiple times per day. A third of the Southern California coast could be deemed one of these urban beaches. If you go out there and look at them, they all look the same. The upper beach is wide and flat, and the intertidal is narrow and steep, and that's not what the beaches normally look like around here. And they're devoid of wrack and plants. In contrast, reference beaches are influenced by natural processes and not subject to these mechanized maintenance regimes. Though both types can be influenced by anthropogenic processes acting at the regional scale that affect the sediment budget, so damming of rivers and things like that. So what are the effects on the biodiversity, structure, and composition and ecological function, and how do these responses differ across the intertidal zone? So for the total community, wrack-associated species, and lower beach species. To approach these questions, we conducted biodiversity surveys at 16 beaches, 9 reference, and 7 urban beaches, from Santa Barbara to San Diego from 2009 to 2015. The reference beaches are in green here, the urban beaches are in black, and I'll use these colors throughout this section to refer to these two types. At least one urban and one reference beach was located within each littoral cell, and those are outlined by the orange dashed lines, and it's a little hard to see. Look better on my computer. We hypothesized that urban maintenance regimes would impact biodiversity and structure, and our results support that. So from top to bottom in the graphs and in order of effect size, the species richness, abundance, and biomass was degraded on urban beaches relative to the reference sites. And you can see a pretty stark contrast from 2 of the beaches that I've taken as examples that are separated by just 15 kilometers. Crystal Cove, we had 30 species. In Huntington Beach, there were only 4. And no wrack species at Huntington Beach. We hypothesized that the negative impacts would be apparent across the intertidal zone, but the impacts would be greater for the wrack-associated species. The left 2 bars are the lower beach, the right 2 bars are the wrack-associated species. Biodiversity and structure were negatively impacted for both the lower beach and wrack-associated species, but the effect size was greater for the wrack-associated species. A group that we know and have been showing are extremely vulnerable to human impacts. Voice is cracking. The composition of species is important as well. So again, we use one of these multivariate approaches, the non-metric multidimensional scaling, to examine community assemblage patterns for urban and reference beaches. Again, each plot represents a beach. The closer the points are together, the more similar they are. They are open open symbols are urban beaches, closed symbols are reference beaches, and they're colored by littoral cell. We found distinct clusters that differed significantly between the urban and reference beaches, circled here. For the total community, lower beach and rack associated species. Our results also showed that community composition differed significantly across littoral cells for the reference beaches, so there were significant differences there. But if you look at the lower beach species, these different they did not differ significantly across the littoral cells. So littoral cells contribute to the biogeographic structure of the region, but the mechanized maintenance regimes has led to biological homogenization of these sandy beach macroinvertebrate communities. And that's kind of an interesting take home that I wasn't expecting on this, for this. So what are the characteristics driving the patterns shown here? First, though, I must point out that urban beaches and the reference beaches look very different if you're just out there. So I'll first talk about the characteristics of the beach and then go into the drivers. So beach width, slope, and Dean's parameter differed significantly between beach types. However, you can see here from top to bottom that rack cover was significantly lower on the urban beaches, and sediment was significantly coarser and less sorted on the urban beaches. We identified the drivers explaining variance in community assembly using the same distance-based linear model that I talked about earlier. We found that grain size and rack cover explained a high percentage of the variability. For the lower beach, it was mainly sediment sorting and grain size, and for the rack-associated species, it was rack cover. So how does that all relate to the ecological function? For our analysis, we use 3 multivariate functional diversity indices: functional richness, dispersion, and evenness. All differed significantly between the urban and reference beaches. Functional richness and dispersion were lower for the urban beaches, but functional evenness was higher because it only accounts for the occupied niche space, and because functional richness was lower, there was less niche space occupied. So overall, these results tend to suggest that ecological function and condition of urban beaches was degraded. So what I want you to take home from this slide is that mechanized maintenance of beaches that's associated with coastal urbanization has very clear impacts on the biodiversity structure and function of beach ecosystems. The effect size was greatest on richness, and we saw stronger effects for the rack associated than lower beach species. The macroinvertebrate communities on urban beaches were altered and more homogeneous. Again, grain size and rack cover explained a lot of high percentage of these differences. And one sort of important take home that's not quite as apparent is that beaches in urbanized coastal areas that are not subject to these maintenance regimes actually can have really high biodiversity structure and function. So despite all of this development and everything, if we are able to still support high diversity structure and function. Almost there. So in conclusion, there are several major points to consider. The community structure of beaches are largely congruent with these, something popped up on my screen, right? Congruent with major biogeographic patterns for the ecoregion. We had these beach-specific variables that were almost as important as the oceanographic variables, so sea surface temperature and salinity and productivity. And importantly, the composition of and connectivity to these donor ecosystems, the kelp forest, affects these biogeographic patterns. The major macroinvertebrate groups responded differently to these processes, acting across these very large temporal and spatial scales. For one, it was a spatial degree of isolation. So rack-associated species are more isolated, probably because of their lower dispersal ability. And there was a difference in the influence of the beach-specific variables. So rack for the rack-associated species and sediment for the lower beach species. And we also saw a difference in the responses to human impacts. Rack-associated species are particularly vulnerable, and it looks like they're losing ground over time. So another take home is that sandy beach ecosystems actually support very high biodiversity, contrary to what other people might think. When you go out there and look, you don't really see much going on there. It's all under the sand. And Southern California represents a global biodiversity hotspot, and that has a lot to do with these really productive coastal regions and huge kelp forests offshore that provide key resources for these animals. These local disturbance regimes that I keep talking about are causing huge impacts on the biodiversity structure and function of these ecosystems. And we were looking sort of for these big patterns and regional trends from climate change and sea level rise, and we saw the substantial decline at this one beach at Colorado Point that were consistent with sea level rise and climate change. And we're likely to see that at a lot more beaches that are locked in that manner. So there's no way for them to retreat landward. It's a pretty big number here, with up to 67% of Southern California beaches expected to disappear by 2100. Our results suggest a need for ecosystem monitoring, management, and conservation. The good news, though, is that our results suggest that there is a lot of potential to restore these ecosystems, but it's gonna take some change and people wanting to make that change and support these important ecosystems. So on that note, I'm on to my acknowledgments here. I think I do okay on time. So I had a lot of funding sources through all of this: Sea Grant, USC and California Sea Grant. The LTR and NSF have been great supporters. Coastal Fund is always big on this, and some of this has been funded by the Coastal Commission. UCSB and IGPMS have been great. Thanks to Live and Dave, Melanie and Azure for doing all of that work. And then on to my committee. Jenny has been an amazing advisor. I can't say enough about what she's done. She took a chance hiring me back in 2007 or so. Almost didn't get hired because of clothing I was wearing, but I was gonna bring that shirt today. But Jenny's something special. She refuses to sit in her office. I think she sampled every beach with me. She was out there regardless of what had happened to her. We went to a beach. She stuck her foot in one of the core holes and a wave hit her knee and she tore her ACL. She still walked a kilometer to count birds and back. And then we had to walk up this really steep bluff. And she was back sampling beaches in no time. This is one day we went down, she didn't have to go, we went down to Scripps. We left at 1 a.m., got down there at 4, a little earlier than we expected. 3 hours from here to Scripps is pretty quickly. And so we had to put some headlamps on and do some work there. She sampled all the beaches up and down the coast. Whenever I need her, she pops in the office or lets me pop in there, whether it's work-related, personal stuff. Yeah, it's been a great 12 years. Yeah, I started as an undergrad in her lab. Steve, I don't know how you do it. You manage like 50 grad students at a time. And while Jenny was my primary advisor, whenever I needed you, somehow you read my papers and gave me great feedback that just, I think it elevated the level of the work that I was doing and helped me step back from beaches. So thank you for all that. Been great. Hunter, I've known you since, Hunter used to live in the house next door to me where I grew up in Berkeley. So I've connected with Hunter as an undergrad, and he promised that he'd take me to Maria, and he never took me. So I got him on my committee, hoping maybe he'd take me, and he never took me then. But Hunter also, like Steve, makes me step back and think more about my research and my questions and step away from beaches. And you've been great on my committee because, again, your soft sediment expertise and all that gives us a different angle. Bob and Mark are also on my committee. Oh, Mark's up there. He didn't want to sit with the committee. Mark, you've been great. I mean, you've been working with undergrads and grad students for years here. I love how you're always deeply involved in everything, and you always pull me back. You're like, don't do that hand-waving thing. Stick to your results. It's always a good addition there. Bob always has been dressing really funny lately. It took him a while to figure out how to get style. Maybe Zoel is giving you tips, but he looked a little Miami Vice here. Again, Bob, you've been great in, there are times that I've been like, God, what the hell am I doing? And you kind of like pick me up and you're like, go have that kid. And you're always a wealth of knowledge, just everything ecology, and I feel like I can always lean on you. Then there's the crab lab. This is not even a complete list. Rochelle was the lab tech when I was an intern, and she secretly trained me in all the beach stuff, so that when she announced to Jenny and Mark that she was leaving, they'd have no option but to hire me, even though I wore weird t-shirts. There are a bunch of grad students and lab techs who have helped out with a lot of this work and just been a lot of fun, listed here. Undergrads, they've gone on to be grad students and done in the time that I was working on the PhD. A lot of us go out to pho on Fridays. I really appreciate that. We sometimes go and we do pub runs on Fiesta and dress in weird costumes and do something that's like a crab pose. So thanks for all that. 518 undergraduates that have come through the lab since I've been there. I think it's more around like 250, but still a lot. They've been a lot of help. A lot of people that I've worked with, mentors and experts. Dale Strawn, thank you for that data set. Dave Hubbard should really kind of be on that committee side if he could. I did lots of long car rides with Dave up and down the coast where we talked about anything from science to family and all sorts of stuff, so I appreciate that. Carrie was one of my early bosses as well. Did a lot of projects with her to help me kind of figure out what I want to do and that I want to go to grad school, and her office is across from me, so I can always talk with her. Dave Stoneman, whenever I have analytical questions about stuff, he's also big on beaches. Has been a big help. Karina Nielsen helped us sample all these Northern California beaches, and her lab processed a lot of the samples, which was a big help. A couple experts there, and I love teaching. I've had some great students. Mark and Bob gave me opportunities to teach these classes. Armin, I think I taught classes for him maybe like 8 or 9 times, all the invert classes. Todd Oakley, they've been an inspiration to me, and I've tried to gear my teaching to the way that they do it. Tom even gave me the opportunity to become an instructor on record. That picture is from a lecture I gave right before Thanksgiving, and there were 12 people there, and one of them was my mom. She took that picture. And the fires hit during it was about this time last year, so Susan and John were a big help in figuring out how to give a final exam when finals were canceled. So that was a good first learning experience in teaching. And then I've worked with tons of TAs and other grad students on stuff, too many to name here. There's my family. My grandma got me interested in marine science. This is, she took me out on a boat. I'm seasick there, so that's why I study beaches. Been messing around in the intertidal on beaches and stuff like that. That's in the Berkeley Marina with my brother. So my other grandma used to read my papers before she passed away and take little notes in the column and ask me questions later. There's my family. They've been great to work with. My dad's no longer around, but he got to, one of the last things I told him was that I got the job working in the lab here, so. So my mom and brother have stepped in and been a big help there. My dog, Lily, She came in late in the process while I was doing a lot of writing and encouraged me to get out on the beach and walk around so I didn't throw out my back or anything. And she loves rack. And she doesn't chase shorebirds. I didn't teach her that, she just, I think, sensed it. And then last, my wife, Katie. I've been joking around that she hasn't known me other than as a grad student, so that picture up there, I think, is like one of the first meetings. She was trying to take a picture with Kevin from The Office, and I jumped in to take the picture. And we started dating shortly after, and I think I had just submitted my application for UCSB, which is why I was at Joe's. She's been there to pick me up. As you know, in grad school, there are a lot of lows, a lot of highs. Here she is wiping tears away as I struggled to get through my vows at our wedding a couple of years ago. And then we brought this recent recruit to the crab lab. That's a picture here. Getting ready to settle. She wasn't showing yet, so I pushed my belly out. And a couple of weeks ago, that little megalopa settled. Devin, she's been quiet here in the corner, and she's been great. It's been hard to focus with the lack of sleep, but she's been waking up like once or twice the last week or two, which has made it a little easier to do this talk. So on that note, I think I hit my time right. Any questions? Yeah, Nick?
Show the pictures, you start out motivating on scales of cascading scales of biodiversity or community, I guess. So this is an example of like very regional communities. And obviously birds come through here. Are there ways that in your view now, like that beaches expand out back to these other scales?
Like to the global scale or regional or?
Yeah, just at like whatever levels that like how does coal oil point fit back into what larger scales does that maybe go back to?
So things like regional patterns and global patterns?
Yeah, it seems like a lot of this is like, rack comes to this place.
Yeah.
Right, and what other instances where the beach goes back outwards?
Oh, so where beaches contribute back to
Back
other?
up the scale.
Yeah, so so they do. We're on the Great Flyway here. You said the birds, so they're collecting a lot of food on the beaches here. A lot of the marshes where they used to get food are degraded and disappearing, so they're relying more and more on beaches for that food source. You also have the recycling of nutrients, so that kelp comes in and it goes back out to shore. We're not sure how much of that supports near shore primary productivity, but could produce some. And you're talking about like just larger global patterns too. We did a little bit of work looking at species richness. I didn't include any of it here, but latitudinal patterns in species richness and getting data from beaches around the world and seeing where we fit. And it appears that there is this peak in this temperate region for beach macroinvertebrates, which you see in some other marine systems, but typically you have a peak in the tropics for a lot of terrestrial systems. That answer the question? Sure. Hunter.
Great talk, very well organized, and very clearly presented, thank you. You mentioned decline in biodiversity at Coal Oil Point and suggested it has to do with sea level rise. But in terms of a more specific mechanism, have you done any analysis to look at the changes associated with grain size or changes in the sedimentary regime?
Well, we did see that grain size does have an effect on the community structure of beaches. I mean, there are things that can't really live in very coarse sediments. There is sort of a pattern on beaches that, the coarser sediment you have gets you this more reflective beach, and you have a steeper slope, and that's generally related to lower species richness. I think things just don't do as well in these coarse sediments.
In a large sense, what's happening with sediments on beaches? Are we less sediment coming into ocean systems, and is that having a potential impact on beach communities?
Yeah, so we're damming the rivers a lot. That's cutting off the source. We're adding sand to beaches that's altering the sorting and the grain size, so we're not really matching the natural sediments a lot. A lot of them are coming from elsewhere, where they mine sand to bring it in. Seawalls are accelerating the movement of sand down the coast. Groins are catching it potentially and changing all those sediment dynamics. Does that kind of answer your question, or? And grooming can also have some effects as well. Yep, John.
Have you noticed any changes in species ranges between your two data sets?
I did some work trying to look for that, but I didn't really see anything. I don't think I worked fine enough of a spatial scale to pick up those types of things. We've done a little bit, it may not be related to climate change, but we went and did a paper with Dave and Jenny where we looked at those upper beach isopods and where they're found now and where they were in the past, and looked a little bit at abundance, and that seems to be more driven by the human impacts than anything else. But we are starting to find some of the southern species, tylos. It hadn't been detected in the past, but it might just not have, people might just not have looked for it. So, yep.
Some of the beaches where you're talking about, where they, again, use the off-road vehicles and such, do you have any feel for what the rate of recovery was for those beaches that have been protected or sort of management?
They didn't seem to recover to their full potential, but they're still in recovery mode, but nothing really about the rate. So over 15 years, over that time scale, they would need to recover fully, I think. And there are some restoration projects going on where there's potential to look at that, I guess. Steve?
So what's been the response of the regulatory system given you've demonstrated, such a strong effect of grooming and other things like that? And obviously there was evidence that it was having negative effects before, but this is much more compelling kind of analysis.
So I don't know if my research has gotten out there yet. It's working, it's getting out there. There's a group of beach managers that meet all the time, and I presented that urban beach stuff to them, and there were some faces in there that didn't seem that happy. And during that meeting, they took us out to show us how they grew beaches at East Beach, which was very weird, and then to give that talk right afterwards. There's a group with, they were really excited about showing us this grooming machine that they had.